Some of it we're, oh, okay. Some of it we're just starting to dip our toes into. So I thought I would share all of it. All of it's unpublished, in hopes that maybe we could have a good conversation. If anyone has some ideas for how we can, maybe enhance these experiments, or if there's other ways in which we can branch off of them. But we're finding some very fascinating data that I'm excited to share with everyone today. So just very quickly, I don't think I have to dive into this too deep with this group, but in general, a lot of my background has been focusing on these three different areas of bone phenomena in order to investigate how cells can be manipulated or signaling pathways can be targeted in order to ultimately help form bone where it's needed, when it's needed. And so I've done some work with development, and a lot of what Vicky does in her lab is development and repair. But also the unique perspective I think I probably bring to the lab is working with Chris Jacobs. We did a lot of mechanotransduction work and looking at bone maintenance. And when I talk about bone maintenance, I mean, so under normal conditions, you have a certain amount of bone mass. And then we all know the classic case of going to space and how you do lose bone mass. But there's also the phenomena of enhanced mechanical loading. And the tennis player is a classic example where their dominant arm, they will form more bone in response to this loading. And so some of the work that we've been able to come up with in part was because of the work that I've had to do with mechanical loading before, and just kind of having a little bit of a unique take on Vicky's mouse model phenotype. So I think everyone is probably familiar with the BMP and TGF-beta signaling pathway family. And just to simplify it today, since there's a lot of ligands and receptors that come together in a complex for these two signaling pathways, it's very, very complicated. But just in general, for anyone who's not super familiar with BMP signaling, there's a lot of receptors that type 1 and type 2 receptor complexes that will come together, bind a ligand, and then you have downstream signaling, specifically for the canonical pathway, phosphorylation of SMAD158, which is translocated to the nucleus. And for our talk today, it will be focusing on how this can enable periosteal cells to differentiate and help in bone formation. And because of, I'm going to simplify even more in looking at BMP2 today, because of this very interesting animal model that Vicky has in her lab. So one of the first publications to come out where we basically removed BMP2 from skeletal lineage cells using the PRX1 Cree. We found spontaneous fractures later in life. And in fact, they start as early as I think we found some as early as P14, but definitely by postnatal day 21, there are some present, especially in the forelimb. And when they fractured these adult mice, you can see in the normal mouse, you get with a stabilized pin fracture in the femur. After a certain amount of time, you get this really robust callus formation and healing. Whereas in the mice where we've removed BMP2 from these skeletal lineage cells, there's no healing whatsoever. And they've confirmed this histologically. And this is the publication if you want to dive deeper into the fracture repair phenotype. So this then kind of leads us to periosteal cells, because periosteal cells are very important in fracture repair, since that's where you get expansion of the periosteum. And then a lot of the cells come from the periosteum to help with repair. So just very quickly, I think we've all seen this diagram a million times, but interstitial growth or lengthening is typically dictated by the growth plate. There are some other factors, but this drives it for the most part. And then appositional growth or widening, which is what I'm going to discuss today, is predominantly dictated by the periosteum. So that's why we're going to be talking a lot about periosteum and appositional growth. In one of my previous studies, Vicky had mentioned I started getting into the periosteum during my PhD studies. This was one experiment where we actually ablated the PRX1 expressing cells in adult mice, and with mechanical loading. So if this was, their tennis player arm, you can actually see that a lot of that response or appositional growth in response to mechanical loading is also lost when you ablate periosteal cell populations. So just diving deeper into what's in the periosteum, we know in the long bone periosteum, there's a fibrous and a cambium layer. The cambium layer is a bit more intimate to the periosteal surface. And the cambium layer was once thought to contain the progenitor and stem cells, but more and more research is showing that few populations in the fibrous layer can also be stem cells or progenitor cells. But ultimately, a lot of studies have been done by different groups to show that these can differentiate into chondrocytes or osteoblasts, and then ultimately become embedded osteocytes in normal growth, but also in fracture repair. And I think Celine Colnat is a good group to look at in terms of characterizing, especially these PRX1 expressing cells. And she's done a lot of work to show that when the periosteum expands during injury, that you get a lot of these PRX1 expressing cells, and that they contribute largely to the fracture callus during healing. And they've also done some transplantation studies with either labeled bone marrow stem cells, stromal cells, or periosteal cells. And she's shown in a couple different publications that periosteal cells are predominantly responsible for these bicortical through and through fractures. And I think Sean Morrison, his group recently just put out a publication showing that if you do smaller, so they did a hole drilling study in a tibia, and if for those more minor injuries where intramembranous ossification is the predominant mechanism for healing, it's actually, they think it's more of a bone marrow contribution for those types of healing. So today, whenever I discuss fracture, it will be the bicortical through and through, where we see mostly a periosteal contribution. So going back then to this animal model that Vicky has, where we've removed BMP2 from the skeletal lineage cells, just a reminder, this is the fracture phenotype where there's absolutely no healing. We then looked at it during development, and very interestingly, it seems that the interstitial and the appositional growth have become uncoupled. Whereas, yes, these mice, they're a little bit shorter in the limb, but it's half the width, which is a very striking phenotype. And this suggests to us that there's an issue with the periosteal appositional growth. And this has been confirmed histologically, where you can see the bones are much smaller. But it's also intriguing that the cortical bone becomes a little bit thicker, almost as if it's filling in at the endocortical surface. I would hypothesize this could be in response to the force imbalance, since the bones are about the same length, but now you have something that's half the width trying to take on the same amount of force just from normal walking. And so it's very possible that they're forming more bone at this endocortical surface to help accommodate that force imbalance. And we've also found that at the periosteal surface, if you stain for that phosphorylated SMAD158, which indicates BMP signaling, it also looks as if BMP signaling is disrupted in the periosteum of these mice.
Emily, could I just interrupt to make sure I'm clear here?
Sure.
This is a knockout that's done in the embryonic stage. This is not tasked to when the PRX1 would be just limited in expression to the periosteum. So you're dealing with a mesenchymal defect here, and I'm surprised that they even grow.
Correct. Yeah. I would agree. And that's why I think the phenotype, part of the reason why it's so fascinating is that they still actually form a limb, and it does seem the shape is exactly the same. And well, other than you could maybe argue with the cortis, the mid shaft, it is a little bit different. But the fact that the most striking phenotype is just the appositional growth is very fascinating to us, because yes, you're correct. We're removing it. It's not an inducible model, so it's removed as soon as PRX1 is expressed.
I'll just chime in and say, Dan, that's because there are other BMPs present. So as you start to remove more and more BMPs, you lose the ability to form bone at all. Yeah. During development.
Got it, Vicky. So basically, BMP is not so important in the early stages. Is that what you're telling us?
Saying in the early stages, it's a combination of multiple BMPs.
Okay, thanks. Helpful.
Yep. Great. Good question. Thank you. And then just also further confirming for collagen type 1 in situ hybridization, we also are losing that osteoblast activity at the surface. So these observations led us to really pursue looking at a periosteal phenotype and what's going on at the periosteum. And we want to know, why is this appositional growth disrupted? And so 3 of the questions we've been thinking about, we've actually have a couple other ideas going on too. And if anyone else has any ideas at the end, it would be great to discuss. But the 3 that I will be discussing today are, we're wondering if the periosteal cells themselves are changed. Is it possible that we're just completely missing periosteal cell population, perhaps a stem or progenitor population? And also, is BMP2 embedded in the matrix important? So just going back to the question that was asked earlier, I think that is a very good note to make, that it's being knocked out in the embryonic stage. So not only are you losing expression in the cells, but you're also losing the BMP2 that's secreted by the cells and deposited in the extracellular matrix. So just going on what we've started so far with this first question, are they changed, and also the second question kind of come together a little bit. Are they missing? We figured the best way to test that would be to do an RNA-seq on the periosteal cells from control and these knockout mice. So far, we actually did try a laser capture to specifically get the periosteum. And there were some complications with that. So we're retrying it, because coming from my previous lab where I did a lot of periosteal cell isolations and actually am able to strip the periosteum off of the bone, we're going to instead do it on the whole periosteum that's stripped from the bone, because it will minimize our processing and things like that. So I've done a trial with this so far. So this is just preliminary data, just to confirm that the process actually went well, and testing our preps before we actually send them to the core for sequencing. So the first thing we did is just do a profile. Obviously BMP2, we want to check that's our positive control, and it's gone. So that matches with what we'd expect. But also, interestingly, there's also a decrease in other BMPs that are important for skeletal growth, and also other ligands associated with the TGF-beta and BMP pathway. And then I have over here on the right, just for anyone who's interested in their favorite ligand or antagonist on the right side, if you want to know, these, we did check everything, but some things were not expressed in the periosteum. We also looked at the full receptor profile, and interestingly, most of the receptors are also down. We then wanted to look more specifically at periosteal markers. One, because we want to confirm that these are periosteal cell populations. Sometimes you can get muscle or connective tissue contamination. And we were able to find that the contamination factors were low, but with sequencing, we should be able to rule out those populations anyway. But intriguingly, a lot of the periosteal markers were also down. And so this is a collection of periosteal specific markers, or highly expressed in the periosteum, as well as stem cell markers that have been identified in the periosteum. And so all of these are significantly down.
Hey, Emily, this is Matt Warman. Can I just interrupt? How many samples have you looked at? Because I'm not seeing error bars on these graphs.
Yeah, so this is just 1 or 2 that I have collected so far. So this is just me testing out the preps and doing some qPCR to validate the preps before we send them for sequencing. So I wouldn't consider this an actual experiment. This is more just preparing for the sequencing where we can fully evaluate all of these genes.
Okay. Are you going to come back, Emily, to why you're getting total wipeout here? I mean, this is a little surprising.
I would agree. And I think it might be, it could be a sensitivity issue. So I think when we can send it for sequencing, we'll get a better idea.
I'd say also that if you look at the phosphorylated SMAD158 levels at this time point, in the absence of BMP2, there isn't any in the periosteum. So that's probably why you're seeing that at this point. It has to do with the very specific expression pattern of BMP2 being the dominant BMP in the periosteum at this point.
I think that's a good point too.
Can I butt in one more time? It's Matt.
Absolutely. Yep.
So I stumbled upon what was, I think, a really fascinating paper that may be of help to you down the road. And it's a paper in which they use this Cree driver FSP1 Cree. It's also called like S100 protein. We have it in our lab, and I think you can also get it at Jack's.
Okay.
And they used it to knock out the type 1 collagen gene in mice. The assumption being that the mice, if you knock out the type 1 collagen, would have embryonic lethality.
Mm-hmm.
And what they found, in fact, is that the mice develop brittle bones only after birth as they age. And for
Oh,
whatever reason, FSP1 Cre is expressed in periosteal-derived bone cells
okay.
postnatally rather than prenatally.
That's
And I think
fascinating.
for you, what would be really nice about that is knocking out BMP2 postnatally.
Mm-hmm.
Already know you can knock it out prenatally and it's not going to harm the mouse,
Right.
using PRX1. And then you'll be able to see a change in the expression, if it does it in the periosteal cells.
Right. Right.
You'll be able to see a change in expression rather than the consequence of not having it from birth.
Mhm.
Exactly.
Oh, there is an ERT2 regulated
Yeah, but
PRX1,
that doesn't, but
which could be
that's not
done
a,
this way.
yeah. But that Cree has a lot of problems, so
Yeah.
decided
I
not,
actually,
not to go that way.
Vicky, we've been using it successfully. I don't know what problems we have, but you have to be a little careful, and but it works for us. Anyway, it's not it's Emily's talk.
yeah. No, this is great though. Thank you. I appreciate the conversation. And yeah, Matt, I will definitely look into that. Thank you very much for bringing it up.
Yeah, we
So,
can show you our data. I think it
okay.
was quite a surprise. And the other thing why I would love to have you look at it is we were thinking, we're having a hard time measuring pain in animals that have brittle bone disease
Yeah.
because they have a lot of fractures. But on the other hand, if we can induce brittle bone disease postnatally, then we could monitor whether they have pain before they get fractures. And your model would be another mouse OI model, right?
Yeah. Right. And it's actually very funny that you bring up the pain as well, because these mice, as they get older, they definitely are hunched and they're not moving the way that they should, but we don't actually see signs of pain in them. So one other avenue we're also exploring is if there's a disruption and innervation in the periosteum, because it doesn't seem that they're actually sensing any issues. So we are currently sort of collaborating with Aaron James's lab at Johns Hopkins and sending out samples. They're going to help us look at some of the innervation in these mice as well. Yeah. So, but yeah, maybe we can follow up another time, and I would love to see the data. Great. Okay. Any other comments or questions? Okay. So one other thing that also has come up in the laser capture I had mentioned initially that we did, but also in these preps that I'm testing out, is there's a downregulation of hedgehog signaling. And so these are classic targets of hedgehog signaling. This is also something we've been able to validate in vivo. So at P0 in the periosteum, you can typically see some of these GLIA1 expressing cells, but it's essentially lost in the knockout mice. And then I also mapped some of these SMAD158 binding elements, and we do find that a lot of the hedgehog targets do have a lot of SMAD binding elements very close to the transcription start site. So it's not surprising that there's probably some interaction with BMP signaling and hedgehog signaling. So unfortunately, we don't have a great way to test this in vivo and in vitro, since if you isolate periosteal cells from the knockout mice, they actually die in culture without having BMP2. And so just coming back again, studying this phenotype really starts to occur at 2 weeks, and we do start to see it also a little bit at 1 week. So I decided to create an ex vivo appositional growth model so we could study maybe supplementing something into the media to see if we can somehow recover this phenotype if we're able to test it out in culture. And so what I essentially did is I started dissecting the femurs from the mice at 1 week of age, and then I give them some mechanical loading for an entire week. And that's because in static culture, we found that the explants were lengthening, but they weren't widening. And just with my background in mechanotransduction, we have done some very cool ex vivo studies, but also just these fluid shear chambers that my former PI actually came up with. So we can shear the cells in culture and look at the effects of mechanical loading. But it turns out we're not actually looking at mechanotransduction in this case, but it turned out to be a very excellent tool for the ex vivo growth model. So just to break it down quickly, in order to keep the femurs in place, because we put them in these plates, add culture media, and then just turn on the rocker, and it moves back and forth, which enables the fluid to shear across it. But in order to keep the femurs in place, I worked with my uncle, who's a mechanical engineer, and he was able to help me design and machine these femur supports. So I'm just going to show you a quick video of what they look like. And so you can basically see in the video, the bone is held in place, and with the rocking, the fluid is able to shear over it. So this allows us to apply some sort of physical loading that would probably mimic when the pups start to move around on their own a bit. And it seems that that mechanical loading is important to really help the appositional growth along in culture. And so I had a dental student who has been working with me. He was able to image these explants live at day, the day that we collected them versus 1 week after we've been stimulating these samples for an hour on the rocker. And he did these little plots, and in static growth, you can see they very slowly grow in width over the 7 days. But if we apply this fluid shear model, you can get a big change in growth. So this model is going to enable us to look at appositional growth and maybe supplement the media or take something out. And it's ultimately going to be a great tool for us to study this. And so just quantifying, also this is then through micro CT, I was able to more precisely measure these. So after 7 days of rocking, you do see a change in width, whereas length really isn't too affected. And that makes sense because length should be driven by growth plate function anyway. And then also the direction of the shear that we're applying, it should be mostly affected in terms of the widening aspect. And then histologically, we can see the periosteum here in the static, and it does expand with this rocking. So using this tool then, using this tool, I was able to take explants from the control and the mutant mice. And if you look at just length and width under normal conditions, if you just look to quantify those on their own, the mutant is always going to be a little bit shorter and thinner. But if you look at the fold change, that really helps us understand the application of the force. So under so length, we don't really see a change, but if I normalize the rocking explant to the static explants, I do see about a 10 to 20% change in width in the control mice. And this is not seen at all in the knockout mice. Looking at this histologically, then you can see for a control, the static is still a little bit larger than the knockout, but with rocking, we really do get a widening in that cortical midshaft, whereas we don't really see a change with the knockout. And so then I was able to take that micro CT analysis a little bit further. And if you look at the cross section, we do see growth in that rocking. And then the knockout, there's no change, but also interestingly, there is some extra growth at the endocortical surface, which we saw in our histology originally when characterizing the phenotype of the mice. So I do think this appositional growth model is working well for us in terms of characterizing these in an ex vivo system. And so if you look at the BVTV, it is also higher in the knockout mice, which isn't surprising because we do see some of that extra bone formation occurring at the endocortical thickness, and they're also thicker. In terms of looking at BMP signaling, so these samples were collected at the end of the study on the left. So I think I do need to redo that because BMP signaling can change rapidly. So I think it would be better to do a time course of these, maybe rock for an hour and then collect samples after that. But at the end of the study, we did still notice that you can see under static conditions, there is some BMP signaling occurring in the cortical bone, but not too much activity at the periosteal surface, and it's just completely absent in the knockout mice.
Emily,
and this
would you,
matches.
would you go back? It looked like, to me, it looked like the PRX1 Cree knockout had greater growth. Did I? Yeah. And then,
okay.
The cortical thickness. Yeah. What's going on here?
So we notice developmentally in our mice that the cortical bone actually gets thicker compared to wild type mice. And I think what's happening is because you have that force imbalance where the length, it's still lengthening about the same, but because the bone is half the width, I think it's very possible that they're filling in bone at the endocortical surface in order to increase mass to withstand just the normal forces of loading. So the purpose of this figure was to show that we get, there's extra bone formation in the knockout mice in terms of having a thicker cortical bone. And I think it's because it's filling in at the endocortical surface.
You know, I think Carl Jepsen showed this for the MOV13 mouse of type 1OI, where their bones are actually thicker. And if you really want to prove that, what you can do is give a dose of, doxycycline followed by alizarin, or calcine followed by alizarin, and then just plastic embed and look at fluorescence. And you'll probably see, as you're saying, no periosteal growth and good endocortical growth.
Yeah, that could actually be really nice, because maybe we would even see periosteal growth in the controls with the rocking, but then we'd see endocortical with the knockouts. That's a good point.
Yeah. So,
and then
what? Let me just jump in, Matt, because I think this is what you and Emily are talking about. So what the trade-off here is, and this is fascinating, I think, is that you're getting, what you're claiming, Emily, is that the endosteal growth is compensating for the periosteal growth. And then I gotta ask the question whether this is gender specific, because that's kind of what we think is a difference between men and women. So this is really interesting, that you would have this trade-off, even with the rocking. I don't think rocking is putting a lot of stress on the bone, but anyway, there it is.
Yeah.
So I find that fascinating, and maybe what Matt's talking about, if you do the labeling, you'll see a labeling endosteal as well as, of course, periosteal, and that would be a way to measure this. Yeah,
I agree. Yeah.
I mean, this is truly not what I would have expected. Did you expect this?
Based on the original phenotype, yes. But I mean, if you would have asked me that when we got the original phenotype, I don't know what I would have said. But just looking at the histology from the original publication, I was expecting it to be thicker. Yes.
How about gender? Do we have gender effects here? Because I could believe that you're going to have strong gender effects.
For this, I don't have it, but that just could be the explant system.
Are these in males or females?
They're in both, but also our the original publication, if we look at male and female mice, we see the same phenomenon in vivo. Yep.
Yeah. The one other thought is it could be that you're not getting new endosteal bone formation, but instead you're getting a failure of endosteal bone resorption in order for the bone to grow. But you'll figure that out with if you do your calcine labeling.
I think maybe Vicky can chime in on this, but I thought we had already looked at osteoclast activity and there wasn't a change in resorption.
Right. Osteoclasts are perfectly normal. We did look at that.
Great. Thanks.
Well, what do you mean by perfectly normal?
See a difference in number between when we look at per surface area, we don't see a difference in number. We have done histomorphometry and labeling.
Okay.
Guys. Yeah. We haven't done it every single age, but we've done it at, I think, 8-week time point. But I have to go back and check.
Sorry, this is Francesca, but if you don't have any resorption and you have endosteal formation like you're thinking, and probably you have, your marrow area should change, right? Because you don't
It
have
does.
absorption, you have endosteal formation. So there is something else that we're missing, I guess.
Oh, that's a good point. You mean like measure marrow area from one time point to another?
Yeah, because here
Okay.
in this image that you have doesn't look like you have any change in the marrow area, right?
Right. Yeah.
So if you're thinking that you increase formation on the endosteal surface to increase the cortical thickness. Yeah.
So Francesca, in vivo, there is a difference in the marrow area.
Yeah, in vivo, yeah.
I think in these short experiments, we don't see it.
Is it not enough?
Yeah.
Okay, thank you.
Yeah. I haven't actually measured it yet. And it's funny you bring that up because David and I were just chatting about it today. So I am going to actually try to measure it because some of these measurements are like this, I think it's pretty, I think most people would say it's reasonable that this, it looks like this is wider than this, right? But these are such tiny, tiny measurements that I would think the marrow area we might need to actually quantify to see if there's a difference. I might not be able to tell by eye. Great.
It actually does look thinner. I mean, it does look smaller in the rock.
You're talking,
Yeah. I mean, you'd have to measure it and you'd have to reduce it in multiple bones, but it's certainly not bigger.
Yeah. Great. Thanks. I'm writing this down. Okay. So then back to just the image quickly showing signaling. We do see increased signaling then with the rocking and the controls, some of it in the periosteum, but also in the cortical bone. And this is just muscle background. And then again, not really any change with the knockouts. I wanted to see if the change in BMP signaling was potentially due to a change in BMP2 expression. We have these BMP2 LAC-Z mice available in the lab. And I did 1 hour of rocking and then I collected the samples. I did a time course and collected them starting 1 hour. Then I did 3, 5, 7, and 24 hours later. And I do see a little, some more staining in the osteocytes, but not really any change in the periosteal surface. And I'll bring up later why we think this is important. So then I had mentioned earlier, brought up this data from my preliminary preps for the RNA-seq, but also the laser capture RNA-seq. We noticed these hedgehog targets and confirmed that in vivo. So one of the first experiments we wanted to try is if we apply a hedgehog agonist. So if we upregulate hedgehog signaling, will this help us recover the phenotype? So I repeated the experiments, and when I took out the explants, when I loaded the explants in the device, I included 1 micromolar of a smoothened agonist. And we do find that, so if you have no treatment, it's what we see normally with width, the controls increase slightly and the knockouts do not really change. But if I add this smoothened agonist, they actually are very similar to the controls. So we think it is partially recovering the phenotype. And we don't, again, see really any change in length with the application of this treatment. So looking at the micro CT cross sections then, so here is the ones I showed earlier. And they are similar to the controls we've had after redoing this experiment. But also if I give it to the control mice, they don't seem to change too much. I think we need to do some histology though, because it looks like the bone that's forming might be a little bit different. But if you look at even under static conditions, the bone is already in the knockout starting to return more to normal. And I would say they're definitely still smaller in size, but the shape of the bone is much more normal and it's getting to the size of the control. So that's why I'd say it's only a partial recovery at this time. So I'd also mentioned too, we're having the part of the reason of establishing this ex vivo system is so that we can study, we can confirm some of the things that we're seeing in vivo and do some more signaling experiments in vitro. So I have actually been trying to generate a periosteal stem cell progenitor cell line, since Vicky's really, really good at making cell lines. I think some people here might actually be using some of the ones that she's created. So I isolated periosteal cells from postnatal day 5 BMP2 LAC-Z pups, passaged them 20 times so far, and then I've cloned, and we have 2 candidate clones that are looking good so far. So I'm just going to go over quickly what I've done to validate it so far, and I'll discuss what we're planning on doing in the future. And then if anyone else who's made cell lines has any comments or suggestions, that'd be great to hear. So the first thing we did is go and look at classic stem cell markers, especially ones that have been identified in the periosteum. And so the 2 clones that I'm really looking at right now, they're just, I don't have a cute name for the cell line yet, but we'll get there. But they don't express PDGF receptor alpha, which is interesting to me because that is an important subset in the periosteum. But they do express these other markers, and clone 1 has very weak expression of CD34. And I'll explain why when I get to the next slide. And then they're also positive for all the periosteal markers. We also made sure they were negative for markers that are unique to muscle, connective tissue, and bone. So we're pretty confident that it is a periosteal line. And then moving on to, I'm going to go over the multipotency first. So I think it's very possible, since clone 1 doesn't seem to want to differentiate along the adipogenic lineage as much as this clone 8 does, and I had mentioned before that the CD34 expression is weak. I'm wondering right now, we'll need to do more studies for it, if it's very possible that this clone 1 is more of a progenitor line, whereas the clone 8 could be more of an actual stem cell line. So we're going to be doing a couple more experiments, repeating these multipotency experiments, obviously. But for right now, we do know at least they will somewhat differentiate into adipocytes, chondrocytes, and form mineralized matrix. We also wanted to check that they're actually responsive to BMP2 and engage in BMP signaling. So I treated with a high dose of BMP2, and we see a very big response. And if you look at downstream signaling targets in these cells. And then also I wanted to confirm that our hedgehog targets are also going up. So we're in the process of doing dosing studies right now to maybe get, since this is very high, to get a treatment concentration that's more reasonable. And we're also going to be looking at how differentiation as well with BMP2 treatment, and how this might be how signaling changes when we do induced differentiation. So that's what we're moving towards right now. And then I also wanted to come back then to the third question that I originally mentioned on my first slide. We want to know what the source of BMP2 is, because I had mentioned earlier, we know that some of these cells in the periosteum express BMP2, and this is in one of the publications. You can see some cells expressing a lot of osteocytes, especially at the periosteal surface also do, and probably, bone lining or cells that are probably on their way to being more of a committed osteoblast also express BMP2. And this is postnatally in these animals. But we're not sure if the reason why we have these changes in appositional growth when you remove BMP2 is if it's a cellular contribution that's driving it, or is it a matrix contribution that's driving that? So what we're trying to set up right now is another tool in the lab where we've come up with this idea. We can take this periosteal cells from the control and the knockout mice and seed them on devitalized bone shafts. So there's no cells in the bone, but there would be BMP2 in the matrix. So we can do different combinations of bones that have BMP2 and cells that have BMP2 with bones that do not have BMP2 and cells that do not have BMP2. So if we were to seed cells from a control mouse on a control shaft, that would be our positive control. We would expect the cells to lay on the shaft and survive. If we seed knockout cells on a knockout shaft, we would expect these cells to not survive. And then we can do a combination where we put cells that have BMP2 on a shaft that does not, see if they survive and lay down matrix. And that will let us know if there is a cell-derived contribution. If we take cells that do not have BMP2 and seed them on a shaft that has BMP2, that will, and again, check for viability matrix deposition, that will inform us if it's a matrix-derived contribution. It's very possible that it's both, but it would be very important to know to complement our work on seeing if there's changes in the periosteal cell population, because it's very possible that sure, there's some changes in the cells, but if you don't have BMP2 in the matrix, they might not be able to differentiate and into osteoblasts and lay down new matrix.
So couldn't you try this using like
Matt, why don't we let Emily finish up?
Oh,
because
okay.
really
Are we
time-
going over time? Sorry. Okay.
Well, you're not going over time. We've all been going over time. So you can
Okay.
take another minute or two, but that's it. But we
Yeah.
really have to stop.
I'm very close. This is actually my last slide, so we can get into the questions on. But yeah, so just this is the technique we've been thinking. We've got this very nice vibrant dye that a collaborator mentioned to us, and I've been able to label periosteal cells that I've isolated from these mice, and then we've been able to seed them on top of these devitalized shafts. So hopefully we'll have some very interesting results from that in the near future. And so I think, yeah, so I think I already talked about my model. And so with that, I'll just go ahead and thank everyone in my lab, especially Gavin and Anna, who are 2 dental students that have really been helping me with a lot of this work, as well as Aaron, who helped with some of the periosteal cell work. And I'll take questions. Ooh.
Matt, why don't you ask your question now?
Thank you. Thank you, Hank. Have you thought about just like using a DMP1-CRE or an osteocalcin-CRE to knock out BMP2 and then see what happens to the periosteum? That might be easier than your devitalized bone experiments.
I think my
So we have used
headphones stopped working.
various CREs to knock out BMP2.
Can you let everyone know?
I'm happy to talk to you about that offline, Matt, but if you knock it out, if you knock out BMP2 in already differentiated osteoblasts, it doesn't matter.
But it's okay.
So wouldn't that argue it's not a matrix protein? It's a periosteum protein?
Sorry about that. I actually just missed a lot of what happened because my
It's okay,
AirPods died.
we don't want to go so far over, but since we're all local, we can talk about this, right? I would say yes and no to your question, Matt, and I'm happy to show you the data. And so the more differentiated the cell is, the less BMP2 matters.
Great. Wonderful. Well, Emily, that was both a terrific talk and a terrific way of responding to almost constant but useful interruptions. So
Thank you.
We kept going with it, and that was great. So now, Tamara, why don't you introduce the second speaker?
Thanks, Hank. And Emily, fantastic job. That
Thank
was
you.
beautiful work. And thanks also for your flexibility and accommodating the schedule change. So I'm really thrilled to introduce you to Dr. Thomas Peterson, who is a core member of the Baker Computational Health Sciences Institute, so a real data science person. And he directs the UCSF REACH Analytics Core, which is investigating back pain. He's also the director of the Laboratory for Digital and Computational Health Science in the Department of Orthopaedic Surgery. And he works with clinician scientists on both prospective clinical trials as well as retrospective electronic health records review. And I have to share that also some really beautiful work on health disparities in our field, which is so important. So welcome, Tom. We're glad you're here and looking forward to your talk on patient phenotyping, levels of evidence, and machine learning for clinical research.
Great. Thank you for that introduction. So hi, everyone. I'm Tom Peterson. So that was a really good overview of the things that I'm doing. And today I'm going to talk specifically about how to phenotype patients, what levels of evidence are there, what kind of data sets exist, and how you can use machine learning for clinical research. So a little bit about me. I'm the director for the Laboratory in Digital and Computational Health Sciences. This is sort of a collaborative environment between the Department of Orthopaedic Surgery, the Baker Computational Health Sciences Institute, the University of California, and the San Francisco VA Medical Center. I am the director of the analytics core for the UCSF REACH for Chronic Low Back Pain Mechanistic Research Center, and I have no relevant disclosures. So just a little bit of background about some of the data that exists here at UCSF. This is the UCSF Information Commons, and this slide's a little bit outdated, but you can tell that we have over 1 million patients, and I believe that's over 1.5 million patients by now. And this is just sort of giving you an overview of the types of data that exists here, right? So you can look at the patient demographics, you can look at clinical images, structured electronic health record data, metadata from radiological images, patient reported outcomes are getting very, very good now, and also concepts that are extracted from clinical notes, right? One of the problems with this this data, though, is that sometimes you need very technical skills to access this data and to study it. And sometimes there's a lack of institutional interest and technical skills required to analyze these resources, right? Over at the UCHDW, or the University of California Health Data Warehouse, this is where all the data for all the University of California resides. So they have 5 major medical, academic medical centers. So here at UCSF, also UC San Diego, UCI, UCLA, UC Davis, and also UC Riverside. So here we have over 5.3 million patients, right? And there's over 1,000 UC affiliated clinical settings that have an electronic health record connected to this data set. And just over here on the right, you can kind of see the breakdown of demographics again, and also where each patient is located. You can tell that green is UCSF because we are servicing the Bay Area and around, but also all up and down California. The ultimate goal of these types of data sets is to be able to connect to larger global data sets, right? What's nice about the Odyssey network, the OHDSI network, is that they have specific data formats to be able to link these data sets together. So these have been standardized here at UCSF and UC-wide. And we're actually working to do some different studies with the Odyssey network, which is in over 18 different countries, 369 million patient records. It is really massive. And so to be able to contribute and do science on this kind of scale is very exciting. However, there are a lot of problems, right? So large scale electronic health record data, it's going to be available everywhere. It's going to be lots and lots, millions of patients that you're going to be able to study. In this case, we're going to talk a little bit about orthopedic surgery interventions today, which is a great problem to study with electronic health records, and we'll talk about why. But there's a big problem because for a lot of conditions, phenotyping, or identifying patients and getting data to study about them and making that data correct is a hard task, right? So one problem is connectivity, right? So there's a lack of interconnectedness between data centers or data silos, right? This can really hinder databases being able to be studied by researchers, and often they're interrupted by clunky data portals. You can lose your password. Sometimes it doesn't work in there. You need to contact an administrator. Can be lots of problems. Clinical images also exist in these databases, but they're not segmented. And comments from clinicians, which is what you really needed to get at, is just in the free text clinical notes, so it's not easy. The structured data exists, and that's probably the most reliable data, but unfortunately, a lot of that data is missing. You know, I probably don't have to tell a lot of people on this call how frustrated people are with electronic health record systems and putting in the right billing codes. There's people whose entire job is dedicated to getting this right, and we still do not. So structured data also has a lot of problems. And lastly, clinical notes. These are stored in free text format, right? So it's very hard to interpret with a machine learning algorithm. This is where really important information lives, like pain scores, the extent of pain, prior treatments. So like have people gotten yoga, massage, or acupuncture? That's not going to be in a diagnosis code, right? And also socioeconomic factors. You know, does this person have a stable living situation? Do they have social support? These types of things, and we're going to go into that. But those are all in the clinical notes, and they're very hard to get at to be able to study. So I'm going to show you two projects that I've worked on in the past to help sort of grab some of this data and make it more usable. So one of the problems with clinical notes is that they're full of identifiable information, right? So they're very hard to actually give to someone to read and pull out the information if you need it, right? So this is a tool called Filter. That's PHI, Protected Health Information Filter. It's So this solves some of the connectivity problems by de-identifying clinical notes, which removes a lot of the risk incurred by sharing the data, right? So this has actually been run on all of the clinical notes at UCSF. They're available in the Information Commons right now. And we've also been able to run a lot of extraction tools, natural language process extraction tools using this de-identified data. And this is going to be available for the whole University of California Health Data Warehouse pretty soon too. So this is an exciting tool that's going to be really useful coming up here in the future. And like I said, we also have some natural language processing tools that we've ran on these clinical notes. So in addition to existing natural language processing tools, so things like CTAKES, which is the Clinical Text Analysis and Knowledge Extraction System, and tools in the Spacey Python library have been run on all the clinical notes, right? So on the left, you can see things that it's pulling out. History of hearing impairment is something that CTAKES is pulling out. On the left here, it's pulling out blood pressure. It's pulling out different chest x-rays that have been done, right? So what these tools do is they go through the clinical notes and tell you what's in there. Now, they're not always right? And they don't always pull everything out equally. So on the right here, this is a tool that we created here at UCSF. This is a natural language processing tool for extracting social determinants of health from free text clinical notes. And these are the categories that we were looking for, right? So looking for anxiety and depression, marital partnership status, pain and disability, which is the VAS pain score, access to housing, social isolation, transportation, access to transportation, insurance status, whether or not they have it, indications of financial strain, and access to food. So this is a metric called F1. You don't really need to know exactly what F1 is to understand this. Just know that if it's closer to 1, it's better. We have 2 different types of levels here that we're looking at pulling out these social determinants of health from the free text clinical notes. But essentially, what you need to know is that we're pretty good at pulling out anxiety and depression, marital status, and pain and disability. But once we get into things like housing, we're still doing pretty well. But on the level 2, meaning specific types of access to housing, we're doing a little bit better, right? So essentially, what this is a way of pulling out this information to be able to use it in clinical research. It still needs some work. In the future, we're going to be applying these models to new data sets and having people go over the free text clinical notes to make sure that we got it right. Okay, so what kinds of data actually exist out there? And let's put it in perspective. What exactly can we use electronic health records for? So there's a lot of clinical data, like what I'm doing over at the Backpack Initiative from NIH HEAL here at UCSF Reach. There's things you can get from existing clinical trials, right? So pooling data together, making collaborators, essentially creating consortiums, things like that. You can get an IRB to look at your current patients, or you can plan prospective clinical trials, right? For electronic health records, there's things like the UC Health Data Warehouse, the Veterans Affairs database is very large and useful as well. And we talked a little bit about the Odyssey network and the different types of data that live in these databases. There's also administrative claims databases. So this is a little bit less than what you would find in electronic health records. So it'll tell you diagnosis codes, it'll tell you patient demographics, and insurance billing claims, but it won't get into a lot of the deep detail, and it won't have any clinical notes like we're looking at. And there's also nationwide survey data, which is more of a population level look at what is out there. And that's even less data than you're going to get with administrative claim databases. So here at UCSF, one of the things we're doing with UCSF Reach is we got a $30 million multi-core prospective clinical trial going on. We're really conducting a wide array of traditional and even novel phenotyping mechanisms for these patients, right? And the idea is we're going to be determining the optimal or most efficacious or cost-effective of these clinical metrics to capture to understand patient-specific outcomes, right? So in comparison to electronic health record databases, well, like I said, we're going to go a little bit into what databases we're capturing first. So first off, there's a deeply phenotyped database called the Comeback database. This is 400 patients. So we have patient-reported outcomes, biobehavioral questionnaires, pathophysiology, so imaging, and also motion capture tests, biospecimen archiving, and these patients are receiving standard of care. There's also a larger data set for the backbone data set. This is a digital cohort, right? So we're not getting as many deep phenotyping elements as we're getting for the comeback data. So we're not getting like imaging and biomechanical biospecimen data. But we're getting shallow phenotyping, right? So this is questionnaires. We're getting all the electronic health record data. They're getting standard of care. We're giving them some interventions like using Fitbits and apps for mindfulness. And compared to the EHR phenotyping, you know, it's even less, right? So we're getting more patients, but we're not going to have some of those questionnaires. We're not going to have Fitbit interventions, mindfulness interventions. We're just getting all the electronic health records, and we also have clinical images and notes, and we have nice tools to process that those data types. But in comparison, the depth of phenotyping for these tools, the comeback is the best, right? So these are deeply phenotyped. Then back home is shallowly phenotyped. And EHR is somewhere in the middle compared to administrative claims databases and nationwide surveys that have very shallow phenotyping. But the benefit of these smaller databases is that they have deep phenotyping, but the larger databases, you have much larger cohort size, right? So the power of your analysis is much better, but you'll have less data to study. And looking at outcome metrics, you're going to have really detailed outcome metrics in these deeply phenotyped patients, like the comeback data set. EHR would be somewhere in the middle, you know, so you can look at surgical outcomes and administrative claims and nationwide surveys are the least. And of course, these have different prices, right? So the comeback data is very expensive to collect. EHR data, while it's expensive to implement, it's now low to free. And these nationwide survey data are very, very cheap or free, right? So the goal here is to understand what data type to use, where and when and why. So let's talk about levels of evidence. So levels of evidence are different levels at which you should accept a clinical study, right, for making clinical decisions, right? So the worst level of evidence would be things that are opinions from authorities or reports from single experts, things like a single descriptive or qualitative study or systematic reviews of descriptive and qualitative studies, right? So sort of meta-analyses here. And then level 4, these would be things like electronic health records, claims databases, and large-scale nationwide surveys, right? So that you have a lot of data, but it's not as good as a clinical trial, right? So moving up the ladder here, level 3 would be things like comeback and back home, right? So these are well-designed clinical control trials, but they're not randomized, right? So we're looking at their standard of care over time. And then we have another trial, which I didn't mention before because it's a little bit newer, but this is actually a randomized control trial that's going to have a level 2 evidence, and it's going to be informed by our results from the comeback and back home studies, right? So in general, you should keep this in mind when you're studying either prospective or retrospective data. Where do the data sets come from, and what is the level of evidence that you can expect to obtain from your studies? Okay, so what do we want to do with this? How do we compare what type of data set to use? So this is sort of a wire diagram of how we're going to use the data. So the comeback data, this is the deeply phenotyped data. It has electronic health records, right? So we can match up to everything that we're getting from the EHR. But it also has this deep phenotyping data like PROs, survey data, quantitative markers, and measurements of physical function. If you're trying to predict the future for a patient, making a machine learning model, what you want to do is you want to divide the data up into 70% for training and 30% for testing, right? So now what this does is this makes a machine learning model to tell you which of these data types are most important for predicting patient outcome. Then similarly, you do the same thing with the backbone data set, right? So there's shallow phenotyping now, so there's less data to actually put in these models. But you create a machine learning model, and you can actually test that machine learning model on the comeback data, right? Because they have similar data that's being captured. You have outcome data that are similar, right? So you can build a model and test it compared to the one you made from the comeback data. And finally, you do that the same with the HR data set and make models using that. Very sparse data here. And you can compare, right? So these are predictive modeling, right? So what are the models, how good are the models using all the data set, all the data? So everything A, B, C, and D from the comeback data set, or going down the line, if we only have E and F for the EHR records, how well does the model actually do, right? Because keep in mind, these are very expensive data to collect. You don't necessarily want to put your patient through a regular MRI or an fMRI if you don't need to, right? So this is a way of actually telling which type of data is the most important for collecting to tell to anticipate patient-specific outcomes. Okay, so in general, there's three main research tracks, as I like to call them, for doing prospective and retrospective research. So the first track is traditional statistical analysis. So these can be things like associating factors with an outcome variable, right? So looking at your covariates and doing univariate and multivariate regression to look for statistical associations with your outcomes. Another thing you can do is look at treatment pattern frequency, right? So look at who gets what, how often people get different treatments, and this can be informative, right? The second research track is unsupervised machine learning, right? So this means we're not necessarily looking at the outcome when we're training. What we're doing is we're looking at phenotypes of patients, right? So looking for principal component analyses or using clustering methods to identify what types of patients exist in the data set, right? So this can tell you, based on different types of phenotypes, whether or not certain patients may or may not respond to treatment, or how exactly to manage your patient population in a sensible way, given the types of patients that you have. And the final one that we're going to talk about a little bit more is supervised machine learning. So this is, you know, the outcome of the patient, you're trying to make a model to predict patient-specific outcomes. So these are things like regressions, decision trees, neural networks. You can get deep learning if you really want to, but there are reasons where you might not want to, and we'll talk about that. And assessment of existing tools, right? Because there's a lot of them out there that already exist. You know, how good are they? How better can you do than them? These are all really important things to look at. So we're going to talk a little bit about supervised machine learning. So no matter what you think of computers predicting the future, this is what they think of you. This is a cute little AI generated image tool. It's trying to generate doctors predicting the future. So as you can see, some of them kind of make sense. Their eyes, faces, and hands don't quite come out right. And the reason that I'm showing you this is not just because it's kind of funny, but also because it's important to know that you might not be able to trust these machine learning tools, right? And the machine learning tool thinks that these are pretty good pictures, and you know it's getting pretty good. But without human eyes on this, you might not be able to tell what makes sense and what doesn't, right? So that's why one of my main goals in my research career is to work very closely with clinicians to be able to add some expertise into these models to make them do better and to stop them when they're going wrong. So some examples of these tools in low back pain, there's tools for predicting recovery from chronic low back pain, transition from acute to chronic, which patients benefit from specific treatments. And like we just talked about, there are a lot of common pitfalls, including increasing clinician burden, which is very bad. I'm going to talk briefly about some tools that exist for looking at spinal fusion surgeries and outcomes here. So for anticipating outcomes, this is very common in spinal surgery. So you can predict how a specific patient is going to respond to treatment, and not just a simple statistical association, right? So this patient, not patients on average. This is a tool for anticipating patient outcomes. So this is the ACS NISQIP risk calculator. This is an online tool. You put in what procedure you're doing in CPT codes up at the top, and you literally fill out these boxes until it describes your patient correctly. And down the bottom here on the right, it kicks out prediction method, a prediction model for likelihood of discharging to rehabilitation facility or likelihood of sepsis. And it tells you a little bit about your risk versus the average risk, right? So this is an existing tool. It's not exactly the best, so you can't, it's not using all the data that you might think is important for every surgery, right? This is the only data it's using, this stuff you're looking at on the screen, right? So what we wanted to do is we wanted to see if we can add additional information from the electronic health record to beat these existing tools. So what we did is we pulled data from the de-identified electronic health record that I described a little bit earlier. We had an exclusion criteria of under 18 years old, if they had missing data, and some other indications that it was not elective spine surgery. Overall, we had we broke it up into cervical, lumbar, and adult spinal deformity patients, and we broke it into a training and testing validation here. And on the right, you can see the demographics. So essentially what we do here, like we were talking about, what we're planning to do with the REACH data, is we split this up into a training and validation set. We're going to use the training set to train models to predict length of stay and discharge disposition using electronic health records, which have more detail than what the NSQIP has available. In the validation set, we go through and we fill out that web form to get a prediction for each patient for the NSQIP prediction model. And then using that validation set, we can actually compare what the NSQIP looks like to our in-house machine learning models. So this is some of our first results here. So what we're looking at is length of stay.
Can I ask you a quick question? So all the data that you're putting in is basically demographic kind of information or yes, no kind of information, not like outcomes of different imaging studies, like MRI or X-ray?
Yeah, so that's a great question. Like I was talking about previously, those images are not easy to get at and they're not easy to interpret, right? This is something that we have, and I fully believe that given some time and the right the right kind of machine learning models to either extract that information from the image or from the free text clinical note, we're going to get a lot better at predicting things, right? But what's nice here is that, like you said, we actually don't have a lot of extra information than what's available in the NSQIP calculator, right? So we have more information on different types of preoperative risk factors, right? We have different types of surgical invasiveness, which is a metric that we came up with for assessing how invasive the surgery is. There's also information related to insomnia and sleep apnea, opioid disorders, which all turned out to be pretty important here. So these are just things that, looking at this NSQIP calculator, it can't even consider. It's not built to consider these things. And, this is built for all kinds of surgeries, not just orthopedic surgery. And so things that you might want for an orthopedic surgery calculator might not be available in this type of prediction model, right? So using this extra data, we were able to get a significantly better prediction of actual length of stay after spinal fusion surgery. And also looking at discharge disposition compared to the NSQIP calculator, we got a much better area under the curve, looking at 0.788 compared to 0.75, although these are not statistically different. But it is slightly better. So you can call that statistically the same or better. And I think that, like I said, once we include more of this, more and more different data types, if we, this wasn't even using natural language processing on the clinical notes. So if you add more and more detailed data, I think you're going to be able to do a lot better than these off-the-shelf calculators. So some takeaways from this machine learning project. This is an AI-based prediction tool. You can anticipate patient-specific outcomes, not just the average outcome for patients. This can actually really help patients anticipate their recovery time or optimize pre-surgery, and it can have an overall beneficial economic impact for them. This framework in general is really nice because it allows us to compare to external calculators like the ACS NISQUIP tool. And we can build models using our data, our in-house data that statistically outperforms the NISQUIP for length of stay, right? And this is only going to get better. You know, the NISQUIP calculator tool and things like it can only go so far. We have all these electronic health records. We're building all these prospective data sets with novel and deep imaging, deep phenotyping. And so our prediction models are only going to get more informed and better. Just some acknowledgements, all my great collaborators, UCSF and the Department of Orthopaedic Surgery, BCHSI, the HEAL Initiative, and UC Health and the SFVA, UCSF Reach. I'd be remiss if I didn't mention they were hiring postdoctoral research scholars. So if you know anyone who has statistical or machine learning skills, send them my way. Thanks.
Thanks, Tom. And I'd love to open up the floor for questions. And maybe one thing I'll just start with is if you can share a little bit more about how you're using these approaches to ask questions about health disparities, especially in the California data sets or beyond.
Yeah, so one nice thing about the natural language processing tool that we created is that this is now available on all UCSF notes, so you can actually look in your data set whether or not these social determinants of health exist, which is something that you wouldn't be able to do using structured information. These types of tools are being pushed out UC-wide, so this is going to be something that's available to the larger community very soon. As far as how you can use this data, there's a huge amount of data, there's a huge amount of research that's being applied to look at social disparity research in general. So looking at who gets care, whether or not social determinants of health or underrepresented populations are receiving worse or experiencing worse outcomes, and how you can possibly mitigate that. And being able to look at this social determinants of health information at the clinical note level, being able to pull it out and actually understand it. We're getting better and better at it, and we're going to be able to really identify ways to improve care in specific ways. Looks like we have a hand up. Daniel?
Yeah, Tom, definitely. So this approach is way out of my wheelhouse. But I have to ask, how, I mean, just gone from the VA over to UC, I mean, we have two very different EHRs, and we can't talk back and forth. So how in the world can you, and now we're throwing in all kinds of different data sets, how do you combine all that? I mean, I'm just sort of blown away by the possibilities. I can see it with UC, you know, the UC campuses, because I think all of those are using the same EHR, but that's not true for the VA, and that's not true for the national VA data set. So how do you do that in simple terms that a guy who spends all his time, working with mice can understand?
Yeah, that's a great question. So there's two answers to that. There's the hard way, which I've actually done before, which is you extract your patient data from one EHR and you put it together in the way that you want it, and you extract your data from the other EHR and you put it together the way you want it. This is very inefficient. But the nice thing is that the Odyssey network is creating a data standard called OMOP. And so this is how they actually got all these electronic health records connected over all these different countries. How the sausage is made really is it starts out in whatever database format you have. So it's in, Epic Clarity here at UCSF. And every different hospital has a different version of Epic Clarity. They have different packages that they buy to make the data set more or less complex. And all that goes into OMOP, right? So there are researchers at UCSF and at these different institutions on all these bullet points that actually go and extract the data and put it into the OMOP format, right? So then you can align all your data sets perfectly because they're in this standardized format. It's my understanding that the VA has something like that. I believe I've seen Odyssey and VA papers that look at their data together, but you know, there are different benefits of doing that as well versus just downloading the data and putting it together the way that you think is right. So yeah, I could honestly talk all day about how specifically this sausage is made, but I think we have a couple other questions, if there's time.
Matt?
Sure. Thanks for the really interesting talk. I have two questions. One relates to how do you deal with multiple hypothesis testing and controlling for that? Because you're collecting so much data and there's so many different hypotheses that you can sort of follow about correlations and outcome measures. And then my second question relates to how do you deal with orthopedic surgeon practices? Because I assume that different orthopedic surgeons have different ways that they like to do lower back pain interventions. And although length of stay in the hospital might differ between the two, they might tell you, but in our experience, 5-year follow-up is different if we do it this way versus that way. So how are you dealing with physician preferences in sort of analyzing these data?
Yeah, that's a really interesting question. So to answer the first question, multiple testing, we do this in a very controlled manner, right? So we don't just go testing whatever we want. We write up a whole statistical analysis plan with specific hypotheses. And if we're testing more than one, then we're going to correct for multiple testing, typically with von Browning. For machine learning prediction algorithms, you don't need to worry about that as much. That's what the training testing split is for. You do that once, and your things that are important in the model aren't necessarily statistically significant, right? They are important for predicting, which is a different thing. And as far as different clinician preferences, in the UC Health Data Warehouse, actually, I was able to publish the first paper that was analyzing this data set. What that paper was looking at treatment patterns in adult type 2 diabetes and looking at who gets what medication when, right? You know, theoretically, we thought originally that there should be some sort of standardized way of doing this. You know, it's diabetes. There's a very regimented way of doing this. And that was very naive. It turns out that all clinicians are different for every possible reason. And one thing that you'll find is that one clinician isn't necessarily comparable to another one, right? So what if this clinician is an insulin specialist, right? So all the people who are already on insulin are going to go to that clinician, right? What if someone here in the orthopedic surgery department specializes in severe trauma? You know, their data sets might look a little bit different than people who are doing, you know, hip fracture repair, you know, and it's not necessarily that one physician is getting worse outcomes. It might be because they're taking on different, possibly harder patients.
Yeah, there was a really interesting talk at a recent GRICE Day, which is the Orthopaedic Research Program at Boston Children's, where their experience is, I think, Staph aureus is the most common cause of osteomyelitis. But when they then asked their colleagues from around the country what the most common pathogen is for osteomyelitis in different parts of the country, it turns out it's different bacteria.
Yeah.
So this whole idea that everything's the same around the United States is not true. And you've now shown the same thing. Everything's not the same, even within the same healthcare system out in California. So yeah, it's gonna make parsing out some of these outcomes more challenging.
Hank?
Thanks, Tom. I really enjoyed this talk. It's very thought-provoking. One of the things I had trouble evaluating, though, was you showed us data about how your input data could predict length of stay pretty well. And I can see how that could be very, very useful for a hospital system, but that doesn't tell me what's useful for an individual patient. For that, I need to know a lot more about the standard deviation of how well you predict for individual patients information that they'll actually be able to use, and how often they're going to be right and wrong. It's a different kind of question. Can you tell us about that?
Yeah, absolutely. So in the textbook reason for why you need these things is preoperative optimization, right? So since this is telling you a specific patient, not a patient on average, is going to have this outcome, you can actually say, okay, well, what happens if you lose 10 pounds? You have this much less risk of going to, say, acute rehabilitation or having this many extra days in the hospital. So you can actually use this as like a what if tool to say, you know, if we optimize a patient in this way, can we actually get better results? And you can even use that to say, okay, we can't really optimize this patient anymore. So that being said, how accurate are we? You know, we're better than the standard that's out there, right? So the NSQIP is the standard for doing this, you know, worldwide, except in specific cases. And whether or not these things are actually adopted and how to get them adopted in a way that will actually improve patient outcomes you're right, it's a different question. You know, do people use NSQIP right now? Probably not that much. Maybe people on the call know a little bit better than me. You know, surgeons and not just you know, hospital management systems. But you know, some of my collaborators, like Sig Bervin, who worked on this with me, he uses these types of tools for preoperative optimization. And if these tools get better and better, you know, instead of having a correlation of 0.55, you could have, you know, closer to 1. As these tools get better, maybe there'll be more institutional trust. And as they get more and more accepted, then maybe they'll be used more frequently.
Thanks. When I look at that NSQIP calculator validation, there sure is an awful lot of spread, isn't there? So that, you know, you can tell a patient that they're going to be in the hospital anywhere from 1 to 10 days. That's not all that helpful to the individual patient. So, but I'm sure that for the health system, and it raises questions for then further feedback and so forth. So I think it's useful, but not quite there yet, it seems to me.
Yeah, and that's one of the reasons, going back to what data set to use and when to use it. You know, something like this might require some more deep phenotyping. Maybe there's something in medical images that might be able to tell us a little bit better. You know, because the doctors, you know, they have that information. You know, they're getting MRIs, they're calculating, you know, RAPT scores, all types of different metrics to assess these patients that aren't necessarily available in electronic health records because they're trapped in clinical notes, or they're in some image somewhere that needs to be interpreted. So yeah, so as we get better data into these things, we'll have better knowledge to be able to make better predictions.
Yeah,
Thanks.
sort of, Tom, just follow up on Hank's concern. You know, you can drive a very large truck through this data, no question about it. But you know, the fracture service that gets us closer to endocrine, and you know, looking at the different fracture services which involve endocrine follow-up, because we like to treat these patients with osteoporotic fractures. So it would be very interesting to apply this type of analysis to that, because it really, in many ways, then affects the care that we as endocrinologists can provide for these people who are coming in with their hip fractures.
Great point, Dan. Tom, I'll ask one more question. I think on this call, we have people who are trying to come up with new molecular diagnostics or markers, new imaging strategies that can reveal disease more effectively on the basic science side. Which of these, what do you think is the low-hanging fruit for what can be incorporated into these approaches next, after the sort of things that are easily entered into an EHR?
Yeah, that depends on the task, right? So someone who's working in immunology might need different things than someone who's working in orthopedic surgery, right? So one of the reasons that we have UCSF Reach is to deeply phenotype these back pain patients, because it's not clear what biomarkers are the best for predicting patient-specific outcome, right? And at the end of this, we'll know. And we'll have a really good idea, right? So having some information beforehand is really helpful in choosing which one. here at UCSF Reach, we're not exactly sure, so we're giving them the battery and of existing and novel phenotyping. Yeah, the best one to use, you choose that by expert experience and how much money you have in your clinical trial.
Thank you so much to both of our speakers and for everyone who joined us. I will joyfully release everyone, especially all of you in Boston, to that perfect fall weekend ahead of you, and likewise to everybody here in the Bay Area. Thanks for joining us. Bye.